Hi learners, in this video I will briefly introduce you to the transportation across plasma membrane. So if you go through this image, you can see that plasma membrane is bilayered structure surrounding the cell. If a solute tries to pass through it, will not be allowed since plasma membrane acts as a barrier to the cell. But certain non-polar molecules easily diffuse through the lipid part of the plasma membrane like the carbon dioxide, oxygen. Uncharged polar molecules like water also diffuse or can be transported through specialized protein channels. The lipid part of the plasma membrane is impermeable to charged molecules, hence their entry is facilitated by proteins, which are also known as channels or carriers, which are specialized for this function and are present as integral part of the plasma membrane. Hence, the plasma membrane is selectively permeable, allowing some solutes to pass easily over the others, hence acting as a barrier. So, the transportation that can be through the lipid bilayer or the proteins can be further divided as passive or active. Further, the passive can be subdivided into simple or facilitated types, and active can be further divided into primary or secondary. So, the passive type works along the concentration gradient. That means solutes moving from a higher concentration to a lower concentration, thereby moving through a gradient. And there is no use of metabolic energy by the cell, which is usually ATP, the cell's currency. Also, the net charge molecules are transported along the concentration gradient and the membrane potential. Now here I would briefly like to explain you the concept of a membrane potential. Now plasma membrane, which is separating the cytoplasmic contents from the extracellular contents, is almost all the time witnessing an in and out of solutes which can be charged. The accumulation of these charged molecules and the overall charge that they carry inside the cytoplasm, and this results in an overall charge difference called as the membrane potential. Hence, the concentration gradient and membrane potential that the net charge has to overcome to diffuse through is called as electrochemical gradient. Now, as I mentioned earlier, passive diffusion can be of simple or facilitated type. In the simple type, the solute diffuses across the concentration gradient without any involvement of a membrane protein. Hence, the diffusion occurs along a relative concentration gradient. The rate of diffusion is correlated to the concentration gradient, how hydrophobic the solute is, and its size. This diffusion is also non-selective in nature, and certain examples would be the diffusion of water, oxygen, carbon dioxide, benzene across the plasma membrane. Facilitated diffusion, as the name itself indicates, the diffusion of solute is mediated by transport protein, which could either be a carrier or a channel. The rate of diffusion here is relatively more than the simple type, and it is selective in nature. Example of solutes diffusing through this type are usually polar and charged like carbohydrates, amino acids, and ions. If we graphically try to understand these two types, we can see that simple diffusion gradually increases with an increase in concentration difference and the rate of transport, whereas the facilitated diffusion attains a high rate of transportation even with the low concentration difference because of assistance of a transport protein. But it reaches a saturation due to a fixed number of proteins available. Here, I would like to mention that many a times when the cell is under stress or when the solute has to be transported at a high rate beyond the saturation, increasing the synthesis of these transport proteins only helps cell machinery achieve its desired equilibrium of solutes. Now coming to the active transportation that can be made against the concentration gradient, it definitely needs assistance of a carrier protein. Since you are going against the gradient, an input of metabolic energy is also required. This further is divided into primary or secondary type. Primary requires a direct metabolic source of energy that can be in the form of ATP hydrolysis or light in the case of halobacteria. The most predominant form of this transportation is the exchange of sodium and potassium ions in the cell across the plasma membrane, which I will cover in a different video. In the secondary transport, an indirect source is utilized where the transportation of one solute is coupled with the other and either of the two are transported by a primary method. An example of this is the transport of sodium ions and glucose in the cells of small intestine that I will also cover in a different video. This diagram summarizes the primary and secondary transport of solutes where proton ions are pumped via a protein carrier utilizing energy from a direct ATP hydrolysis. These ions are further pumped back in the cytosol across a concentration gradient which is coupled with the solute S that is being actively transported uphill or against the concentration gradient. Secondary active transportation can be further divided into symport or antiport types, where in symport both the solutes are moving towards the same direction and in antiport both are moving in different directions that could be either intercellular or extracellular. And with this, I would like to conclude this brief video on transportation across the plasma membrane. Thank you for watching and please let me know your reviews on this lecture in the comment section. Goodbye.